Welcome all to the today's class. So today we'll continue with our embryology module, and it is the systemic embryology, that is development of various organ system we are going to see. So we will discuss about development of the kidney. We begin with our quote, master MRCOG with concept oriented clarity. This is just the recapitulation of the knowledge what we have learned in our first MBBS. Okay, so when it comes to the development of the kidney, what is the origin and embryological basis, and from which layer? So whichever organ which we are discussing, from which layer, whether it is coming from ectoderm, endoderm, or mesoderm, that is what is the most of the time question in the your exam. Okay, so germ layer, it is the intermediate mesoderm. Okay, it is the intermediate mesoderm. We have seen that the embryonic mesoderm is divided into three, that is paraaxial, intermediate, and the lateral. Okay, so the lateral further divides into splanchnic and one more. Okay, so this intermediate mesoderm is uh, uh, takes part into the development of the urinary structures, including the kidney. Okay, so the intermediate mesoderm, urogenital reach, gives rise to urinary structures, including the kidney. Nephrogenic cord, the portion of the urogenital reach that forms the successive kidney system. Okay, so it is the nephrogenic cord that is the portion of urogenital reach that forms successive kidney system. There are three successive kidney systems. Okay, so human embryonic kidney development progresses craniocaudally. Okay, it develops craniocaudally through three systems, that is pronephros, mesonephros, and metanephros. That is the definitive kidney. Okay, so the pronephros or forekidney, it appears around 4 weeks in the cervical region of the intermediate mesoderm. It forms the nephrotones and pronephric duct. It is non-functional in humans and regresses rapidly by around 4 weeks. Okay, so pronephric duct persists and forms the foundation of the mesonephric duct. Okay, then second is a mesonephros or entering kidney. It develops caudal to the pronephros at roughly around 4 to 8 weeks. It consists of mesonephric tubules connected to the mesonephric duct, that is the Wolffian duct, and temporarily it is functional. Early in fetal life, it produces small amounts of urine. Okay, so the entering kidney, that is mesonephros, is temporarily functional in early fetal life, but this also regresses. Okay, it produces small amount of urine. Then mesonephros also largely regresses, but in males, mesonephric derivatives form epididymis, vas deferens, seminal vesicles, and ejaculatory duct. So here they can ask you the SBA. The epididymis is derivative of which part from the embryological structure. So it is the in males, mesonephric duct will give rise to epididymis, vas deferens, seminal vesicles, and ejaculatory. In females, most mesonephric remnants will regress. Then coming to the final, that is the permanent kidney, that is metanephros. It begins development at around week 5 and is functional by approximately week 10 to 11. It originates from two key structures, that is ureteric bud, which is the outgrowth from the caudal mesonephric duct. Okay, so from the caudal portion of the mesonephric duct, you get an outgrowth, that is the ureteric bud. And second is a metanephric blastema, that is the condensed intermediate mesoderm. So the intermediate mesoderm will have condensation in a particular area, that is called as metanephric blastema. So these are the two different origins for the development of the kidney. So how it progresses further? Okay, so there is a reciprocal induction. So there is a reciprocal induction. This ureteric bud will induce metanephric mesenchyme. Okay, and that will lead to the branching morphogenesis, and it will lead to the formation of the ureter, renal pelvis, major and minor canalises, and the collecting ducts. Okay, so this the reciprocal induction of the by the ureteric bud causing metanephric mesenchyme, and that will lead to the formation of ureter, renal pelvis, major and minor canalises, and the collecting duct. Now, this metanephric mesenchyme directs the ureteric bud to form nephrons. Okay, so what does the metanephric mesenchyme do? This metanephric mesenchyme will direct the ureteric bud to form the nephron. So, ureteric bud will influence on metanephric mesenchyme, and metanephric mesenchyme will have the directives on the ureteric bud, and it will form the nephron, that is the glomerulus, leading to the formation of the proximal tubule, loop of Henle, and the distal tubule. Okay, so the loop of Henle develops from the from the ureteric bud. Okay, it develops from the ureteric bud. Okay, So this is what we have seen. So we have pronephros, and the metanephros. See, this pronephros will regress over a period of time. Okay, and this mesonephros also regresses to large extent. In males, it will form certain structures. Okay, so this is what we have discussed, and there are two things: this ureteric bud and this metanephric mesenchyme. These two portions, this metanephric mesenchyme and this ureteric bud, which is arising from the caudal portion of the mesonephric duct. Okay, so this is an outgrowth arising from the caudal portion of this mesonephric duct. Okay, so which is called as ureteric bud, and there will be reciprocal induction. So this will influence on this and will lead to the development of certain structures, and this will influence on the ureteric bud and will lead to the development of the nephrons. Okay, so then there is a kidney ascent and rotation. So initial formation occurs in the pelvic region, kidney ascent to lumbar region, it will rotate medially and will get revascularized. So what are the clinical correlations? So congenital anomalies of the kidney and urinary tract. Okay, there can be renal agenesis, which is because of the failure of the ureteric bud or the metanephric mesenchyme interaction. So if this reciprocal induction or the ureteric bud and metanephric mesenchyme interaction is not there, that will lead to the renal agenesis. Then horseshoe shaped kidney because of the fusion anomaly. Then there can be ectopic kidney because of the failure of the ascent, and knowing embryogenic origins aids in the understanding of these anomalies. So it is very important to know this particular thing. Okay. Now we'll see how the bladder develops and the ureters develops. So what we have seen is the development of the kidney. Now look at this particular figure. So this is the cloaca. Okay, so it will it is a common opening at the end of common opening for this allantois, as well as it will form the urogenital sinus also and the caudal part of the hindgut also. So urinary bladder develops from the cloaca. Okay, this cloaca will form the urogenital sinus. It is a part of the hindgut derivatives. Okay, so at 4 weeks, endodermal hindgut ends in a cavity called as cloaca. So this hindgut has got a has got an endodermal cavity called as cloaca. This cloaca is a chamber for hindgut, allantois, and the primitive urogenital ducts. So these are the three things which are opening into the cloaca. That is the hindgut. This is the allantois. You see this allantois. This is the terminal part of the hindgut, and this is the cloaca. Okay, so three things are there in the cloaca. One is a hindgut, second is a allantois, and third is a primitive urogenital ducts. Okay, so then there is a development of the urorectal septum. Okay, so one septum develops called as urorectal septum, and it divides the cloaca into certain portions that we'll see in the next figure. Okay, so a urorectal septum grows caudally and divides the cloaca into anterior urogenital sinus and posterior anorectal canal. Okay, septum meets the cloacal membrane and forms the perineal body. Okay, so this perineal body is coming from this urorectal septum. So this is the urorectum. So this is the part of the cloaca. There is a formation of this urorectal septum. This urorectal septum will meet at the level of the this membrane. Okay, cloacal membrane. Here will be the cloacal membrane. There it ends, and the anterior part will form the urogenital sinus. Okay, and posterior part will form the anorectal canal. Okay, so this urorectal septum will divide the cloaca into two portions. One is the anterior urogenital sinus and posterior anorectal canal. And this urorectal septum will meet the cloacal membrane and it will form the perineal body. Okay. Now this anterior urogenital sinus, what are the parts? It has got a vesical part which will form the urinary bladder. Then it has got a pelvic part and the phallic part. Pelvic part will form the prostate in males and the urethra in females, and the phallic part will contribute to the external genitalia. Okay, so this urogenital sinus, see, this is the urogenital, this urorectal septum will divide the cloaca into anterior and posterior region. This is the urogenital sinus, and this will be anorectal region. This will form the urinary bladder over a period of time. So it has got a vesical part which will form the urinary bladder. It will have the pelvic part which will form the prostate and urethra in females, and it will have the phallic part which will contribute to the external genitalia. And this is the anal membrane. Okay, this urorectal septum will come here and will meet this cloacal membrane. Okay, so that is what is the development of the urinary bladder. Okay, So same thing is shown here. This is the cloaca. This cloaca has got urogenital sinus. This urogenital sinus will lead to the further formation of the bladder. Okay. So that is what is shown here. And these are the mesonephric duct. Okay. So this mesonephric duct will come and will get have the opening into the initial part of the cloaca, and later on your ureter will also have the opening into the bladder. Okay, so how does this bladder gets formed? So bladder arises from the upper part of the urogenital sinus. So origin of the bladder is from the endoderm of the urogenital sinus. Okay, so this urogenital sinus is from the endoderm. So the bladder is coming from the urogenital sinus, and the urogenital sinus is from the endoderm of the this particular from the endoderm. Okay, so initially continues with the allantois. Allantois will later on becomes the urachus. Okay, this is also SBA. What is the fate of the allantois in the adult? It is a urachus. Okay, so urachus will lead to the formation of the median umbilical ligament. So this will form the median umbilical ligament in the adults. Okay, so early bladder has got opening of the mesonephric duct and the ureter. So it will have the opening of both mesonephric duct and the ureter. Then there is a ureteric reimplantation. As the bladder enlarges, the ureter will migrate cranially and laterally, while the mesonephric duct will get incorporated into the bladder. So this bladder will have the opening of the mesonephric duct, as well as ureters. Okay, so it will have the opening of both. But what is going to happen? This mesonephric duct will get incorporated into the wall of the bladder, while the ureter will migrate cranially and laterally. So final ureteric orifices are located separately from the Wolffian ducts. Then there is a histological maturation. The bladder epithelium is a transitional epithelium. This is also SBA. What type of epithelium you get in bladder? It is a transitional epithelium. Okay. Muscle layers form from the splanchnic mesoderm. That is the detrusor muscle. Okay. So that is the origin from the mesoderm. Okay, splanchnic mesoderm will give rise to the muscle layer. So muscle is coming from the mesoderm. Okay, while this most of the bladder is coming from the endoderm of the urogenital sinus. Okay, now let us see the anomaly. So if there is a patent urachus, it is because of the failure of the urachus closure. Okay, there can be urachal cyst, that is because of the partial closure. Then there can be ectopic ureter because of the abnormal ureteric incorporation, or there can be vesico-ureteral reflux because of the incorrect ureteric angle. Okay, so these are the abnormalities which can happen in the development of the kidney and the bladder. Thank you.